The overall goal of this procedure is to study the response of human immune cells to respiratory syncytial virus or RSV infection. This is accomplished by first culturing an infectious RSV stock on HeLa cells. In the second step, the cultured RSV stock is purified by ultracentrifugation. Next, primary human blood immune cells are isolated by density centrifugation. In the final step, the isolated immune cells are stimulated with the virus. Ultimately, the cellular immune response to RSV infection can be assessed by ELISA, qPCR, or flow cytometry.
This technique is a valuable addition to study respiratory syncytial virus infections. RSV is highly adaptive to humans. Therefore, it's important to use human immune cells to study the inflammatory pathways involved in the pathogenesis of the virus.
Although the virus used in this video is RSV, this method can also easily be adapted to study other viruses, such as influenza virus or rhinovirus.
Begin by warming a vial of human RSV seeding stock in a 37 degrees Celsius water bath. When the virus has thawed, dilute the stock in 4 ml aliquots of infection media per flask, keeping the MOI at 0.1 or below. Add the virus to the HeLa cells in as low a total volume as possible without causing the cells to dry out, and then incubate the cultures for 2 to 4 hours at 37 degrees Celsius and 5% CO2, swirling the flask every 15 minutes to redistribute the medium. Next, remove the inoculum and wash the cells once with PBS. Then add 15 milliliters of culture medium to the flasks and incubate them for 3 to 5 days under the same culture conditions. Observe the cells once a day every day.